Okay, welcome back, everyone. So today we're going to continue our walk through the nephron. So last time we were talking about the first part of the nephron, the renal corpuscle, but specifically this relationship between the glomerular capillaries, so this network of blood vessels that are being that are in contact with Bowman's capsule, that first part of our tubule network, and the pressures that push and pull fluid and, along with that fluid, other solutes into the nephron, potentially on their way to become urine. So now we're going to start talking about processes that help us adjust the contents of the urine. So we're going to be thinking about how can we move potentially additional things into that tubule later on. But before we get to the bladder, what processes move stuff that we've filtered into the nephron, but really want to keep in our bloodstream. So we got to reabsorb it back into the body. Okay. So we left off with this slide about reabsorption, showing us in total how much of the fluid, so that water up top, and other substances in the plasma, and therefore in that initial filtrate in the tubules, gets reabsorbed back into the body. So we can see that there's a lot of reabsorption that's going to need to happen here. Lots of things very near 100%, and other things still above like 50% in this table. And one of the ones that we're going to focus on as an example is glucose, because you can see that's our highest number here. Glucose is usually reabsorbed 100%, and that's usually happening in the first part of the nephron in the proximal tubule, which is what we're taking a look at next. Okay. So when we're looking at the proximal tubule, we've moved a little bit in location, right? So we started out here. This is our glomerulus and Bowman's capsule, right? So we're following our filtrate, so our future urine, into this first part of the nephron. So that's our proximal tubule. You can see that the blood flow has also continued, right? So coming out of that efferent arteriole, we can see that the blood continues wrapping around and staying close to the nephron. So first in these peritubular capillaries. So capillaries right next to the tubules. So when we talk about reabsorption in the proximal tubules, we're thinking about that relationship between this first part of the tubule network, or I guess second, who's counting, this proximal part, so once it actually looks tube-like, and that blood flow that's remaining close to our nephron. So this part of the nephron, this proximal tubule here, right? And this one goes down. Usually we are drawing it kind of side to side, but that depiction doesn't really matter for our purposes, right? But this first part, proximal part, is where we're going to see most of our reabsorption happening. So this is where we take tons and tons of water and all those solutes, all those substances that got just kind of bulk filtered into the nephron, and we try to immediately readjust, push a bunch of stuff back into the blood. We already know we don't want that to end up in our urine. So 70% of the sodium and water that we reabsorb and take back into our bloodstream is going back into the blood here from the proximal tubule. And 100% of the glucose that we're reabsorbing is going back into the blood here in the proximal tubule. So when we look at the cells that line this region, we're going to see that they look a very kind of specific way. Okay, so we can see how up here on top, it's kind of fuzzy looking. It's got all these fingers, right? So this is our apical membrane, apex meaning top. This is the part that's going to be facing the fluid, so facing the filtrate, facing that future urine. And what all these projections are a bunch of microvilli making up our brush border, right? So you can see how it looks kind of like a hairbrush up at the top here. So just a reminder about how we name sides of a cell, as this is going to be kind of important as we're looking at these diagrams. Right, the bottom here, this part is our basolateral membrane. So lateral, sneak up a little bit up the sides. And that's where our bloodstream is going to be. So those paratubular capillaries are going to be on that flat side. Now, when we look at how the cells are attached to each other here in the proximal tubule, we're going to notice these little kind of coffee bean looking things on the image. Right, so these are tight junctions. We usually think about tight junctions as forming a solid seal across a layer of cells, across a membrane. But these tight junctions here in the proximal tubule are going to be a little bit leaky. So we are going to see some things sometimes sneaking kind of between our cells here in our proximal tubule. So that's what paracellular transport is. Paracellular is going to be between those cells. So some of that's going to be possible here in the proximal tubule. Okay. So when we're thinking about reabsorption, right, our direction is from the tubular fluid into the bloodstream, right? So we're going to cross that apical membrane. We're going to cross the body of the cell here. We're going to cross our basolateral membrane. There's a little region here of fluid, our paratubular fluid, kind of like our interstitial fluid when we're talking about our generic capillaries, before we hit our capillary endothelial cell and end up in the plasma, so end up back in our bloodstream. And we can see some of the things on this diagram that are going to be really important for driving this reabsorption in the proximal tubule. So I want to point out to you already here that we have sodium-potassium pumps on that basolateral membrane. If any of you took bio of the cell as your prerequisite for this course, you may have looked at some cells that looked a lot like this, tracking how our sodium gradients work, right? But we can see separately, even if you didn't, with this sodium-potassium pump on the basolateral membrane, right, what we're setting up. We're taking all that sodium, pumping it out. So we're going to see that we have high concentrations of sodium here in our paratubular fluid. We have low concentrations of sodium here inside those tubular epithelial cells. And we'd have relatively higher concentrations of sodium here inside our tubules, inside the nephron in this tubular fluid. All right, so we've talked about this way back in A&P 1 at the beginning as well. All right, we set up a gradient here where sodium is going to want to move into the cell down this high-low gradient. That's our favorable direction. That's our downhill. Right, and then we're using energy to move sodium from low to high over here across the basolateral membrane. And we can tell from this image that this is actually how we're pulling glucose into the cell, right? So we take this sodium gradient, creating our kind of power here for glucose reabsorption, right? So our energy use was in this first step, setting up our sodium gradient. So when we move glucose into the cell along with sodium, right, we've got our example of secondary active transport, right? So here we've got primary active transport, active transport. Right, so that's step one. And then our secondary active transport here. Those will be written out in words later on in the slides, but picture tells 1,000 words, right? So this is what this picture is telling us. So this is one of the ways that we're going to see reabsorption happening here in the proximal tubule. Okay. All right. So we're going to be using our example of glucose to think about something called a renal threshold. So to understand that, we want to think conceptually about what this reabsorption kind of does here. So thinking about that image on the previous slide, when you imagine that sodium glucose transporter on the apical membrane, the one where the secondary active transport was happening, imagine it has like kind of a time frame it works. Imagine it takes 1 second for that transporter to work to move glucose into the cell. Okay. Now imagine that earlier on in the nephron, where we have that glomerulus coming into contact with Bowman's capsule, imagine that we have 3 glucose coming out of the glomerulus into Bowman's capsule and heading down to the proximal tubule every second. Right? So what do you think is going to happen if we've got 3 glucose swimming by this single sodium glucose transporter? Okay, let's take a look. Okay, so we got kind of split decision here, but conceptually, what we want to think about, right, as we're walking through our nephron, right, so glucose coming out of the glomerulus, moving its way into the proximal tubule, we're going to see that those transporters that exist, they're kind of limited in how much glucose they can move. They do have kind of a time frame like this. They have a maximum speed that they can work at. So the only way you'd be able, conceptually, to move more glucose would just be to have more transporters overall. We also kind of looked at that a little bit in A&P1, kind of when we were starting metabolism stuff. We talked a little bit about glucose transporters. Right. But if you don't have enough transporters, they're kind of full. They're doing as much work as they can. Right. This means that that glucose just stays in the nephron. It just keeps zooming on by, passes the proximal tubule, ends up in our next part of the nephron into the loop of Henle. Okay. So ultimately, as it continues down the nephron, it's supposed to get absorbed in the proximal tubule. If it doesn't, it's going to go to your bladder and end up in urine. And this is why an old-timey test for diabetes was to test whether the urine was like sweet, because this is what's going to happen if you have too much glucose in the plasma and you have more than your kidneys are able to reabsorb. Okay. So that's the conceptual idea of our transport maximum. Okay. So our transport maximum is going to be that rate of transport when these transporters, which we might also call carriers, are fully saturated. So they're fully occupied. There aren't any extra. Every single one of them is already moving a glucose across the membrane. It's busy. Okay. So that's our saturation of these carriers. And so we have a way to express what the amount of a solute in the plasma is that gets us to that transport maximum, right? You want to know, like, okay, so if we're seeing glucose in the urine, right? Like, imagine we have sweet urine here. What does that mean about the blood glucose, right? Like, how could you back solve, figure out what people's blood glucose levels are, right? So you can say, okay, we know how much glucose we are able to reabsorb with the number of transporters that there are. If there's extra coming into the urine, that means that the plasma has extra glucose in it. We're seeing more glucose moving into the kidneys than we can actually handle. Okay. So that's our renal threshold, right? So our renal threshold is the amount of solute in the plasma that causes an excess and causes this spillover into the urine. So our threshold is the total maximum amount, basically, that we can handle from the blood. So glucose reabsorption is really helpful for thinking about renal thresholds, partially because diabetes is common and partially because 100% of that glucose is supposed to be reabsorbed under normal conditions, right? So there's no real complicated math we have to do here. We don't need to think about later parts of the nephron. So that glucose is getting freely filtered at the glomerulus. So it's just coming straight through into your kidneys in the same proportions, those same percentages that we see in the bloodstream in the plasma. Okay. All of it is supposed to get reabsorbed in the proximal tubule. So move from the proximal tubule back into the bloodstream via those paratubular capillaries. And there shouldn't be any in your urine. Okay. So those carrier proteins for glucose reabsorption, we looked at in the image, right? Across that apical membrane, we had that secondary active transport with sodium, which we set up by setting up the sodium gradient. On the basolateral membrane, once you have glucose being actively pulled into your cell, right now we have a high level of glucose in the cell. So when we're headed towards the blood across that basolateral membrane, we can just diffuse. We do need a protein to go through, so it's facilitated diffusion, but we can go down our high-low gradient easily because we've created this high concentration in the cell. Okay. So we're looking at this again, right? So here at the bottom, I won't circle it this time, is that sodium-potassium pump setting up our gradient. But we can see that secondary active transport happening here at that apical membrane. So we can see that glucose has a red line. So that means this is the unfavorable direction. It's going low to high, right? Which means that there's high glucose here in the cell. We got this green line across the other side because that's our passive direction. We can see there's a protein involved. That's this little circle, right? So that's what's happening on the basolateral membrane. Okay. So typical level of glucose in your blood, in your plasma, is 100 milligrams per deciliter. And that's the number we used as we were calculating out filtered load last time. We just multiply across, basically, multiply our amount of glucose or any other substance in the plasma times that glomerular filtration rate. So times like the amount of fluid, basically, that's carrying that solute through, which is how we get to a filtered load for glucose of 125 milligrams per minute at that 100 milligrams per deciliter value, because we had 100 milligrams per deciliter, which we convert to milligrams per milliliter, multiply it by our 125 milliliters per minute, which was our typical glomerular filtration rate, to get this 125 milligrams per minute, our filtered load of glucose. I'm going to make you recalculate that out now, but if you want to see those steps, they're in our kind of previous section of stuff we went over on Monday. Okay. So this is the amount of glucose we have headed into the proximal tubule under normal conditions. Okay. So this process of taking glucose into our proximal tubule epithelial cells here, right? The number of transporters or carriers that we have here can handle 375 milligrams per minute of glucose, right? So under normal conditions, right, we can see 125 milligrams heading past the proximal tubule, not a problem at all, right? 375 milligrams per minute is way higher, right? So we can totally take all of that glucose into our renal cells and then into the plasma here, okay? So when we think about this, convert it over, right, back to our deciliter values, right, we get a theoretical renal threshold, so a blood content of glucose in the plasma, right, of 300 milligrams per deciliter. So this is the amount that your plasma glucose would have to get to before we equal out that transport maximum. All right, so if we back solve here, we'd get to this as your total value of glucose that you can handle. So that's what a renal threshold is. A renal threshold is that blood value that can be totally cleared as we pass that proximal tubule. Okay. Now, when we actually look at kidney function and when we actually look at glucose reabsorption, we'll find that that's our theoretical renal threshold, right? So that's it. Absolutely every carrier of glucose gets saturated. This is the maximum we could handle. But when we're thinking about how a tube works, right, imagine the glucose is literally swimming through a tube. Those carriers are embedded in the walls of the tube, right? So they're embedded in the cells lining the tube. They do actually have to come into contact with those carriers in order to get pulled through, right? So this means some of them at really high levels of glucose are probably going to sneak by, right? Some of those glucose molecules are going to be just like kind of right in the middle of the tube and they just don't happen to knock into the edges. And so they can actually pass through the proximal tubule without getting reabsorbed, even if there happens to be one carrier somewhere that isn't full, isn't saturated at that moment. So when we look at the actual levels of plasma glucose that your kidneys can fully reabsorb, we see a much lower number here. So the actual renal threshold in practice is about 160 to 180 milligrams per deciliter. And if you took that and did the same calculation for filtered load that we do up top, that would mean about 225 milligrams per minute of glucose starting out in the nephron. Okay. So when we are thinking about these numbers, right, filtered load is our amount of glucose that is in the nephron. Our transport maximum is the amount of glucose we can move here per minute. And our renal threshold is our blood value that gets us to that transport maximum. So these are all kind of similar concepts, especially this transport maximum and this renal threshold. This is specifically about the transporters. If we're thinking about the transport maximum, and if we're looking at the renal threshold, that's going to be a blood value. Okay. So when we look at how glucose gets handled by our kidneys, we can look at our plasma concentration of glucose. So if you measure your blood, how much glucose is in it versus how much glucose is getting moved, okay? And we're looking at three different kinds of movement in this graph. So we're looking at filtration, right? So this is going to be glomerulus into the proximal tubule, right? So from the blood into the nephron, I might say. Okay. Our reabsorption, right? So that's how much glucose is moving back into the bloodstream. So this is going to be from our proximal tubule into our peritubular capillaries. Okay. And excretion is moving out of the body. So this is basically in the urine. So out of the body. Okay. So we're looking at movement in each of those kind of ways on this graph. Okay. So we'll start with the filtration. That's the one that's the straight line, so easiest, right? So what we see is that as your plasma concentration of glucose goes up, right, we got a kind of one-for-one relationship, right? So add more glucose goes into the nephron, gets filtered in. Easy, simple relationship. No adjustments necessary here. Okay. Now, if we look at the reabsorption, that's one that we can see is a little more complicated, right? So that's where our transport maximum and our renal threshold come into play. Okay. So what we see happening here is at first, as your glucose goes up, right, we can see initially we have that same one-to-one relationship that we saw with our filtration because 100% of that glucose is getting reabsorbed into our peritubular capillaries, going back into the blood. But when we hit our renal threshold, we can see something changing. So they've drawn this line for renal threshold at the actual observed real renal threshold rather than at our theoretical one, right? So we can see it in that 160 to 180 range here. Right, so this is where we start to see reabsorption kind of failing, right? So we start to see not quite 100% of that glucose getting reabsorbed anymore. Still getting filtered, but not reabsorbed. So now, right, we've got a difference in these amounts of glucose. And on that third line, we can see where it's going, right? So this is how we start to see spillover into the urine for excretion, right? So anything that doesn't get reabsorbed stays in the nephron, continues out of the kidney as urine. Okay. So we can see that at the renal threshold, this true renal threshold, this observed renal threshold is when we start to see that happening. And we can see that when we hit the transport maximum, which is that maximum amount of glucose that the carriers in the proximal tubule can move, right? When we hit that transport maximum, right, this is where we see a total plateau, right? So when we hit that transport maximum, there's absolutely no possibility that we could move any more glucose into the blood. All our carriers are saturated. So then any extra glucose that is getting filtered in, all of it gets excreted. So that's why we see as this line flattens off, this excretion line starts to match that straight filtration line because every bit of that excess is getting peed out. So that's how this kind of works in practice when we're measuring these values. So you can use this to track blood glucose. You could also, in general, or also potentially, let's say, use values like this, not necessarily of glucose, but values like this for other substances to measure how well the kidneys are working, right? So when we think about reabsorption and filtration and excretion, one of the things we're sometimes trying to figure out is the nephron functioning appropriately? Good. So when we're looking at that reabsorption, we're looking at that return of molecules that were filtered out from the glomerulus into Bowman's capsule, are returning to the blood, so coming out of the proximal tubule and going into the peritubular capillaries. So hopefully all the glucose is being reabsorbed. Hopefully our blood glucose levels are in that normal range. So below our renal threshold, below that transport maximum. And we definitely have other substances that we want to reabsorb as well. Major one is water. We know about 180 liters of water are getting filtered into the nephron every day. You do not want to pee out 180 liters of water per day, not possible. Only maybe 1 to 2 liters of water are usually excreted as urine, could be even less, right? So this means we have to do a lot of water reabsorption as well, right? So a lot of this water reabsorption is happening here in the proximal tubule, right? So when we look at urine contents, you are going to see them go up when you're well hydrated. You are going to see them go down when you're dehydrated, but we are still, in either case, reabsorbing a bunch of that water. Okay. Minimum amount of water that we need to kind of leave in the nephron to continue into our loop of Henle, our distal tubule, our collecting duct, and then send down to the bladder is only 400 milliliters. Okay. This is because you can't just send like chunks of salt through the nephron. They need something to move through. So this 400 milliliters is that minimum amount that can pull the waste products along through your kidney and out of your body. So this is what we think of as our obligatory water loss per day, right? This amount is going to come out no matter what, depending, kind of regardless of how hydrated or dehydrated you are. Otherwise, we start to see weird stuff happening with all these waste products not exiting the body. Good. So we're reabsorbing lots and lots of water. Most of it is getting reabsorbed in the proximal tubules. We'll see that the loop of Henle also does some water reabsorption. There will be some more complex details there. But when we're looking at the proximal tubule water reabsorption, which is most of it, this is unregulated. So we're going to see no hormones involved, no nerves involved. This is just water moving kind of on its own out of the proximal tubule. Okay. I say on its own, and that's sort of true, but it is going along with ions, right? It's not going along with a hormone. It's not like getting directly pulled, but there's stuff moving along with it. There's stuff changing the gradients of our tubular fluid, of our filtrate, as well as the contents of the plasma. So as a rule, we know from our understanding of osmosis, water likes to follow solutes, right? If you drag a bunch of solutes, you drag a bunch of salts towards the blood, water is going to try to water them down. So osmosis is going to pull water along in that same direction. Okay. So this is why our primary solute that we'd be interested in thinking about when we think about water reabsorption is sodium. Right? So we saw sodium is being pumped out across that basolateral membrane. And we have the sodium-potassium pump moving lots of sodium towards the paratubular fluid and then ultimately towards the bloodstream. Right? We're doing that actively through that primary active transport. We're setting up this sodium gradient that's super useful to do secondary active transport across the apical membrane of things that are important to us. But that means that functionally we've created this sodium gradient that water is also going to follow, right? So through osmosis, we're going to have the water moving towards those higher levels of sodium on the other side of the cell. So that sodium that's building up in the paratubular fluid right next to the bloodstream is pulling water towards itself. So we think of sodium formally as establishing an osmotic gradient for water reabsorption, meaning we created a high concentration of sodium that pulls water towards itself following our principles of osmosis. So we can see how that's happening. That active transport of sodium towards our bloodstream pulls water towards it. And we'll see it's not actually the only solute that's moving in that direction. So chloride ions are actually moving along with that sodium as well in that same direction. So we have NaCl, table salt, all kind of getting moved back towards the blood here. Okay. So when we're looking at osmolality, so when we're looking at amounts of solutes versus water of the various regions of our nephron and of our kidney, which we'll do in detail when we look at the loop of Henle, so in some of our later sections, we're going to be comparing them to the osmolality, so the concentrations of solutes pulling on water in our bloodstream. Okay. So when we are looking at that pull on water in the filtrate versus pull on water of our blood plasma, when we're at the glomerular capsule, that first part of the nephron, that Pac-Man looking piece, we have an isoosmotic situation, right? So solutes are roughly the same concentration in both locations there in that very first piece. But as we move through the proximal tubule, we're passing through these regions where we've actively used energy to pump sodium across the basolateral membrane into our paratubular fluid and then sending it into our plasma. We change this situation, right? So now we have higher concentrations of solutes on the other side of the cell. So in our blood plasma, we're raising the osmolality, we're raising the pull on water on the opposite side of the cell, which means we have a force to pull water across by osmosis. I'll have a picture of that in a little bit. Okay. So when we think kind of where this is happening, we looked at our locations. So we have tight junctions between our cells, kind of a little bit more towards the top of the cells, towards that apical side. We do know that they're going to be a little bit leaky, which will come into play when we're looking at the water. We know that that apical side is also really good at reabsorbing, in part because it has so much surface area from that brush border, which is made up of the microvilli. Right? We have the sodium coming into contact in pretty high quantities with those microvilli, with that brush border, because it's got lots of surface area, and being moved into our cells here of the proximal tubule pretty quickly because we've created a low sodium concentration inside the cell by actively pumping sodium out on the other side across that basolateral membrane. Okay, so overall, our sodium is moving straight across the cell towards the blood. Okay. Now, this means that as we're pumping sodium, which has a positive charge, towards the other side of the cell, so it's going to be ending up here in this paratubular fluid first, as soon as it crosses that basolateral membrane. Okay. We've got this positive charge building up here. This is actually going to have an effect on chloride ions because we know positive and negative charges attract. Right? So this isn't so much a gradient thing as it is an electrical charge thing. Negative chloride ions are going to be attracted to sodium on that basolateral membrane side of the cell because of this movement of charges. This means we have kind of double the force for water to move, right? So now we had sodium ions on the opposite side of the cell pulling on the water. Chloride ions came and joined them because of those opposite charges. So now we have both sodium and chloride ions collecting on that blood side of the cell. So in the paratubular fluid, right next to our plasma in the capillaries, right? So now we've got an even stronger force for water to move past those tubular cells into this essentially a modified interstitial fluid, this paratubular fluid, and then ultimately end up back in the bloodstream, back in the plasma. Okay. All right. So I am just going to mention what this picture is doing. Sorry, they labeled a bunch of things with X and Y, so it doesn't map great onto exactly these individual solutes, but it's worth noticing. This first image is showing some of the theoretical ways that we can move something across the tubular epithelial cells for reabsorption. So you could do active transport here at the basolateral membrane, use that to create a gradient. I would do passive transport at the apical membrane. You could do the reverse. You could have active transport at the apical membrane, allowing for passive transport up here at the top. Down here at the bottom, they'd have added on water. So if you do this and end up with high concentrations of solutes, potentially sodium and chloride, over here in the paratubular fluid, that creates a big force for water to get reabsorbed across this cell. And in general, we can have other substances potentially following this same kind of pathway. You can see water is happening, getting reabsorbed kind of near the side here. So this is indicating a little bit of paracellular transport, but we could conceptually move all the way across the cell as well. Okay. I don't know why they labeled a bunch of things with no real ions as examples, but they did it again here with a couple more details, right? So this one, at least, they showed us that sodium-potassium pump, right? So that's driving our sodium gradients. So we can see that sodium gradients can pull stuff along with them, that secondary active transport, right? Once we have a high concentration of a solute here in the tubules, we can let it passively go across the other side. We've still got this X where we have active transport of some other kind across the basolateral membrane, working kind of the same as that establishment of the sodium gradient. And here we can see our water following all these solutes that start to build up in the plasma on the opposite side of the cell, right? So I'm getting reabsorbed by osmosis. And we can have other solutes that kind of follow this same direction. So we'll talk very briefly about urea later, but this is another solute that gets reabsorbed passively, kind of in total, kind of like the water does. So in general, we're just thinking about lots and lots of reabsorption. That's mainly what I'll expect you to know for the proximal tubule. When we think about details of the individual carriers, I'm not going to say we're never going to mention them again, but those are really going to be more important when we get to the distal tubule and the collecting duct, where we see regulated reabsorption. So don't get too worried about all these specific cellular pathways in the proximal tubule. Okay. Now, one of the things we do want to make sure that we know, right, when we say our proximal tubule is our mass reabsorber and that we're seeing all this unregulated reabsorption, right, we want to make sure that we're keying in on that unregulated bit and that we know how that's going to be different later on compared to regulated reabsorption that we see in the distal parts of the nephron. So let's make sure we're there and we know what this unregulated piece means. Okay, let's take a look. Okay, yeah. So when we say unregulated, I want to make sure that we all know unregulated is just about the signals that are causing reabsorption. So unregulated means you're not going to be changing it with your brain. You're not going to be changing it with hormones. When we did walk through those pathways in the proximal tubule, one of the things that we did want to notice is there was some active transport happening, right? So unregulated doesn't mean there's no pumps, doesn't mean there's never active transport, doesn't mean you don't use energy, just means you don't have a signal coming from elsewhere in the body, okay? All right. Well, that's our reabsorption, right? So we've followed some directions, right? So we had filtration from our glomerulus into Bowman's capsule and then into the rest of the nephron. We had reabsorption, lots and lots of it happening, especially as we move stuff from the proximal tubule into our paratubular capillaries, so back towards the blood. Excretion is hopefully kind of obvious. We'll have a slide on that in a second, but that's out of the body. So the last thing we want to make sure that we are adding onto our vocab list in terms of direction words is secretion. So secretion means that we are moving stuff from our paratubular capillaries into our nephron, right? So secretion is any movement from the blood to these tubules. That happens later on, right? So not at the glomerulus, but anywhere else you see stuff moving from the blood into the nephron, that's secretion. Okay. It's crossing all the same cell layers as reabsorption. So we're crossing the epithelial layer of our nephron. We're going to cross basement membrane, crossing paratubular fluid, crossing a capillary endothelial layer, but ultimately in the plasma, right? That was our reabsorption kind of layers. Secretion is the same, just the opposite direction, right? We're coming out of the capillary, crossing its wall, then crossing the wall of the nephron, moving into the tube. Okay. A lot of the transport mechanisms would be the same. So you'll see active transport, not all that much passive transport coming to the top of my head, right? But like all our typical kinds of transport, active, primary active, secondary active, could potentially be helping us drive this, right? What we secrete is going to be a little bit more limited than all the things we filter, especially. So when we look at secreted substances, we mentioned, as we were talking about acidosis and alkalosis already, one of the big things we might want to track are hydrogen ions. We know that that's going to be related to pH, right? So we can adjust how much hydrogen we secrete into the tubules, and therefore how much acidity we remove from the blood eventually. But we got other substances that get secreted too. Potassium is probably going to be the other big one for us in this unit. But as you move into future courses, you might talk about choline, you might talk about creatinine, you might talk about medications like penicillin. There are a couple other things that your body can secrete. Okay. Okay. So we've got a table from our book. Just want to bring to your attention in case you want to make sure that you're being thorough, knowing what can get reabsorbed, what gets secreted, and where. So we've split out the nephron by segment. So proximal tubule, a loop of Henle will go through next, distal tubule collecting duct. You can see there's a lot of stuff that gets reabsorbed, right? That secretion bit is really limited. So you can see the only two things we're really focusing on for normal, average, everyday kidney function are those hydrogen ions and a little bit of potassium here. Okay. So secretion is important, but way fewer things to remember. Okay. Yeah. Right. So when we're thinking about reabsorption, we will also want to think about other locations. So most of our reabsorption and all of our unregulated reabsorption is happening in that proximal tubule. But our distal tubules and collecting ducts are also going to be really important for the ultimate concentrations of especially fluid that we see in the urine, and therefore how much fluid, how much water is staying in your bloodstream. Okay. So when we look at the later parts of the nephron, we're going to see regulated transport happening across the epithelial layer, so across the wall of our nephron. Okay. And we're going to see slightly different structures, right? So if you look at the cell layer here, you can see that apical membrane. This brush border isn't really pretty, right? So it's not got those nice projections of microvilli. It's just a little fuzzy looking. When we look at our tight junctions, those little coffee beans, you can see they're really close together here, right? So these are really tight junctions. So these are limiting our paracellular transport. So we're not going to have stuff sneaking between our cells here. Okay? And this means that we're going to be able to force stuff to go all the way through a cell, which is why we're going to be able to regulate movement across this cell layer with hormones. Then we can just zoom in a little bit on that. We're naming the sides of the cell the same. It looks really similar, but a little bit of a modification. Okay. We'll pick up there next. We just got a couple slides left here before we jump into looking at water overall as a concept.